focused on immunogenicity and worries around that, and if we should worry or not. We'll have Professor João Gonçalves, as I was telling you, is professor at our pharmacy school here in the University of Lisbon. We collaborate for many years. We have shared several experiences, several talks, several training sessions, and also several papers. And I'm sure that you will enjoy. Okay? As usual, after that, if you have questions, we can have a discussion in a mixture of English, Spanish, and with the help of one mediating the process if needed. Okay?
Thank you very much, Ron. I'm sorry, my Spanish is completely 0%. It's not so great, so I will talk in English in a slowly fashion, and I hope you can understand. So what I'm going to talk, what João was asked me to talk about, was a little bit about, the title was this Dissecting Immunogenicity of Biosimilars, and I put Biologics and Biosimilars because this is like something that we hear a lot on biologics. Biologics are proteins, and the immune system tends to respond. It tends to react against proteins. So there is always this question, if we have a biosimilar that comes from another factory, comes from another process of manufacturing, if there is any change somehow in triggers of immunogenicity, innate immunity, or other triggers of immunogenic response that happen with the biosimilar that did not happen with the original drug. And this is important because we don't want to have different immunogenic responses for the biosimilar compared to the original drug. So we don't want to give patients the capacity to react to a drug that it was not done before for the original drug. So I will talk about immunogenicity in the first part, which is more at the basic level, just showing what is important for immunogenicity to occur. How can we measure immunogenicity? What's the implication of that? And then switch to the biosimilar part and just try to give you some data about this, data from the literature and also our data, also in collaboration with Professor João Henrique. So basically, to start about immunogenicity is to talk about pharmacokinetics, right? So we are cared about immunogenicity. We care about immunogenicity because we basically don't want that these curves of pharmacokinetics are altered. This is the main reason for that. So if you consider that the immune system is the most sensitive way to detect differences, so if you have a patient giving the same drugs, but in a different context, or even with some kind of quality alteration, the immune system will detect it. We know this from the last 20 years with biologic drugs and so on. So we can detect those differences at the immune system level. So we need to understand first what is happening with the original biologic drug, and then the biosimilar. And we need to be not so much qualitative, but also very quantitative in the way that the immune system reacts against the two drugs. So at the end of the day, what we want is that these curves here are basically not altered when you switch from a patient which is on an original drug from a biosimilar. So we have here, for example, the pharmacokinetic curves of infliximab. We have here etanercept. We have here ustekinumab. So basically what is important here is that you understand what is happening with what we call what the body does to the drug. So this is a normal curve. So we need to understand this when we deal with immunogenicity. Because at the end of the day, what will happen is that if the patient reacts against a biologic drug, some of this, this is what your patients are having, and basically going to have a maximum concentration, and then you're going to have like this AUC, which is the total extent of the drug, of the patient to the drug. So if you have immunogenicity, probably this will be altered. And then there is this phase of absorption. Of course, for infliximab, we don't have absorption. because it's an IV drug, but for adalimumab, we're going to have these differences here. So it's important for us to understand that. And also we have the Cmax, we have the distribution phase. And when we have the distribution phase, this is very important because all these phase one clinical studies, they assess all these differences here on the distribution and elimination. Why? Especially on elimination, we have this distribution and elimination, we have the binding of antibodies to proteins. And this binding to these proteins, which are receptors that exist in a patient, if there is an alteration in the binding of the drug to these receptors, then we're going to have differences in elimination. And this is what we are looking for. It's that we don't have this. Okay, so we want to maintain really the curve and the clearance. We don't want that the clearance will be lower. So this is important when we look at immunogenicity. This is basically what we are looking for. Is differences this is like the biomarker of immunogenicity. We don't want to have differences at this level. So when we look at dose-response relationship, when you give this dose to a patient and then you want to have a response, basically you want to, in the middle here is the blood concentration. And the blood concentration is really this relationship between pharmacokinetics and pharmacodynamics. So I will talk about immunogenicity because this is the most qualitative, quantitative way of analyzing differences between two drugs. But what is important here is that immunogenicity is well correlated and should be correlated with pharmacokinetics. We cannot discuss data of immunogenicity without looking at the data of pharmacokinetics. And this is something that the research papers and papers that we see on this meta-analysis usually don't do. So this is something that we should stress for the next avalanche of biosimilars, is that the immunogenicity really relates to this blood concentration of the drug. So the therapeutic index is basically what is in the middle. I mean, we have the maximum concentration, lower concentration, I have those curves. Basically, this is what we call the therapeutic index. And many factors contribute to this therapeutic index. But the immunogenicity affects all of these factors. So all of these factors that are important for the clinician to have the right effect for the dose that is giving, all of this is affected by immunogenicity. So this is why it is important for us to look at the data of immunogenicity, probably not as a clinical decision point, okay, we will discuss that, probably not as a clinical decision point for treating the patient, but is important for comparing different formulations, biosimilars, biologics, pharmacovigilance, whatever. So it's this evaluation that we make on the immune system that is important to characterize these drugs. So what are the consequences of immunogenicity? They're going to have, and we know this from the literature, okay? We have differences in safety, of course. Safety is important. They neutralize endogenous counterparts and unique functions of other proteins, hypersensitivity, infusion reactions, efficacy, and all of that. So basically, this is what we are cared about immunogenicity. Okay. So of course, we have data about that. We know that ADA, anti-drug antibodies positivity, reduce this anti-TNF response. We have this meta-analysis from Sander Garcia showing that if you have antibodies in rheumatic patients, what you're going to have in rheumatologic disease, you're going to have here a reduction in the anti-TNF response. We also know that there is a sustained remission by DAS28 that correlates with absence of ADA. So if you have anti-drug antibodies, we have this difference between the clinical effect of patients with and without antibodies. What is interesting here is when we see this percentage of patients we develop antibodies, and if you look at 1 year, 56, 54, 56 weeks, when you look at this, basically the majority and the major probability of a patient to develop immunogenicity occurs during 1 year. So this is why the clinical trials to assess immunogenicity should take at least 1 year. So 1 year is the time that we need at least to have a good sensitivity of analysis to detect antibodies against drugs. drugs, original biologic drugs in a context of a new drug, or even when you compare a biosimilar with the original biologic. And of course, this also depends on methotrexate. I'm sorry, the previous one was adalimumab. This one is also adalimumab. So there is this difference in the use of methotrexate and the reduction in the anti-drug antibodies. Of course, different immunosuppressor drugs have different capacities to neutralize this development of anti-drug antibodies, like for example in gastroenterology or rheumatology. So the critical concerns also about safety is that when you extend or reduce the half-life of a product, it can influence its toxic properties. When you have antibodies against a drug, you may redistribute the drug for other sites that did not see the drug before. So when there is the formation of anti-drug antibodies, there is the formation of immunocomplexes. And so there is this redistribution of a product to different sites that potentially may derive safety implications. And the presence of these complexes, immunocomplexes, can have these physiological consequences. So this is very important because we have data really showing in meta-analysis that the presence of antibodies against a drug really derives more safety events. So what is anti-drug antibodies? So basically when you have like a chimeric antibody, which is infliximab, so you know that the name of the drug when it finishes by iximab, it means that the drug is a chimeric drug. When it finishes by zumab, it means that the drug is humanized, and umab is a totally human antibody. Actually, if we have more time, I would like to tell you the story of adalimumab, because it's not really a fully human protein, but this comes to another meeting. But the interesting part here is that when these drugs come to a patient, there is always this response of the patient with antibodies against sequences that are not fully human, that the patient are not tolerant to that. So basically this response is to the idiotype. Idiotype means that is the region here that binds the antigen. That's why the antibodies, when the patient responds with antibodies against a biosimilar or a biologic, it neutralizes the function. Why? Neutralizes because the antibodies really bind to the place where the drug binds to the target. So it's basically on that. So these high affinity antibodies really are what we call persistent positive responses. And this persistent response is like when we detect antibodies more than 2 times, more than 2 months apart. Okay? So these are persistent positive responses. But also this immunogenicity can be transient. And transient means that some patients respond with antibodies against a drug, and then on the second or third administration, we don't see those antibodies anymore. And those are what we call transient. Transient because they are not really stimulated and held by the T cells, meaning that these antibodies are not high affinity antibodies. So when you look at your patients, basically you are worried about this persistent and development of this persistent high affinity neutralizing antibodies. So the kinetics of ADA development is also very important. And I told you before that 12 months is probably the most sensitive way that we have to analyze the development of immunogenicity. So 12 months is probably the most sensitive. Why? Because usually the clinical effects, they don't appear in the first 12 years. But we now, with the advent of biosimilars, we have good diagnostic or good technologies, more sensitive technologies to detect the appearance of antibodies, both binding antibodies, transient antibodies, and sustained antibodies, high affinity antibodies, at 6 months. So before, the data that we had is that you should have only this assessment here. 12 months, 12 months was the most sensitive time to assess the development of antibodies. Now, with the biosimilar studies, with the technology that the companies are putting to assess the development of antibodies, we know that at 6 months, 4 to 6 months, we already see the development and we can assess the detection of these antibodies. So now we have a problem with immunogenicity, because the problem is the assays, the immunogenicity assays. So many companies, before the advent, and this is very interesting, because before the biosimilars came to the market, all the papers that were talking about immunogenicity against adalimumab, against infliximab, all of them use different assays to detect anti-drug antibodies. And this is a problem, because we cannot compare the development of ADAs with one assay compared to another. So if you want to compare data of immunogenicity, you have to compare with the same assay that was proposed by the study. So usually now we have several studies. We have the standard ELISA, we have something that we call bridging ELISA, and we have more sensitive ones. Companies now are using this luminescent assay here, ECL. It's more sensitive, but usually now in the clinics, the majority, 80% of the people, or 90% of the people in the world use this bridging ELISA. So bridging ELISA is probably the most spread type of assay to detect antibodies against drugs. But the companies that are developing antibodies, they don't use this basically. They use this electroluminescent assay, which detects even more antibodies in a more sensitive way than the bridging ELISA. But I think that the bridging ELISA is pretty enough for the clinical data, for the clinical decision or evaluation that we have these days. So how can we detect? This is just a figure just to show what is happening when we detect antibodies. We basically have the patient develop these anti-drug antibodies in green, and these are the drugs. Okay, so we have drugs in a plate, and then we put the serum of the patient, and then we detect it with a labeled drug, and then if there is this bridging, that's why it calls bridging ELISA. If we detect this signal here, means that the patient has this green antibody. So at the same time, we can detect also these neutralizing antibodies. So this is a technology that this is quite spread in the world. We can detect IgM and IgG. Neutralizing antibodies is not so spread. We do it in our laboratory, but it's not so common to assess neutralization of these antibodies against drugs. But we have another problem, just to give you some problems now. Okay, so the question is that immunogenicity is very important, but we need to be careful with the data that comes from there. And another thing is that if you want to detect the presence of infliximab or adalimumab in the presence of anti-drug antibodies, we have a problem. Because there is already the formation of immunocomplexes, and when you want to detect the drug levels, the drug is not there anymore. So we have to treat the sample with acid. The acid will separate the immunocomplexes, and then we can detect the drug. The same for ADAs. So if you have a high concentration of the drug, and at the same time you have anti-drug antibodies, you cannot detect these anti-drug antibodies. So we need to have some kind of a trick, a laboratory trick, to assess that. That's why for a clinical decision, there is a lot of variability in the immunogenicity data that comes. So that's why the pharmacodynamics, safety, and efficacy helps to determine these clinical consequences. So these are some optimal screening assay questions or characteristics. Be sensitive enough to detect any level of specific antibody in a patient's serum. Okay, so the assay must be very sensitive, have no false negatives, and the false positives that it might have, it must discriminate between what is positive, what is negative, and be reproducible. So we have, before coming to the clinician, before coming to a clinical study, before analyzing this in a biosimilar study, all of these must be very well studied. Okay, even a clinical immunology lab that tries to assess immunogenicity should have all this in place. So this is, for example, one example what we do in the lab. So when we want to assess the immunogenicity against a original biologic drug or a biosimilar, we have the screening assay. If it is positive, we do a second confirmatory assay. Okay? So we don't trust on the first. We do a second one. And then we characterize that assay and we assess the neutralization. Okay? So this is important because I know that many hospitals use commercial kits, and sometimes they don't do all that. And many of these results sometimes are positive when they probably are false positives that are coming. So we usually confirm all of that. And I think the companies do that because the EMA and FDA request that on the guidelines. The companies, when they submit the data about immunogenicity on phase 3 clinical trials, they have to submit the data and say how they have done the immunogenicity assays. And this is important to understand how they have done it. So it's not probably false positives, it's not false negatives, it's really the true evaluation of immunogenicity. So detection of a lot of confusion around, okay? So detection of antibody formation against therapeutic antibodies, there are different assay formats, no standards, drug interference. So it's probably important for characterizing and assessing drugs, but for clinical decision, I have my own doubts, and we can discuss that. So limited importance of ADA for clinical decision. So if we have a lot of variation here, we need to have more biomarkers to compare the ADAs for clinical decision. And I think the pharmacokinetics and some kind of pharmacodynamic markers should be included in the clinical decision together with the ADA. But don't get really dependent on the ADAs for your clinical decision. This is important for characterizing drugs and assessing different responses against the drugs in a group level, but in an individual level, you probably have these clinical parameters that are more important for this assessment. So now we go, after telling you about what is immunogenicity, how we can assess immunogenicity, what are the factors that influence immunogenicity? Because this is important now for the biosimilars. So when the companies are trying to develop biosimilars, they have to be careful with the drug. So we have several characteristics, those that are associated with the drug that influence immunogenicity, those that are not associated with the product, basically associated with the way that the drug is given to the patient, and also associated with the patient, meaning with the pathology, with the disease. So associated with the product is something that we should be concerned for the biosimilar. Here is probably more important when we try to assess the clinical studies that were done with the biosimilars and comparing biosimilars with the original drug. So T-cell epitopes is one of them. T-cell epitopes means that the sequence of the amino acid sequence of one drug compared to another is different. If those are different, then the T cells will recognize peptides presented by the MHC, and they will respond with activation of T cells. But biosimilars is one of the conditions of biosimilars, and I'm sure that we have heard this before yesterday. The sequence, the amino acid sequence of an original drug and the biosimilar is 100% equal, okay? So there is no different T-cell epitopes when you compare one drug to the other, okay? So these are equal between the two drugs, self, non-self is the same. Also, post-translation modifications, there is a lot of discussion about that, but the quality of and the capacity that the companies have to assess these drugs today, really, we can assess all these modifications. We understand which modifications are important for the biosimilars. But something which is very important is here the formulation, impurities, and aggregates. Why? Because when a drug comes to the market, it's not only quality of the drug does not finish when the drug comes out of the factory. It is also very important in the hospital, okay? When the hospital prepares the drugs, when the hospital pharmacy gives the drugs to the patient, the quality of that drug is very important. The stability of that drug is very important. Why? Because if the drug is not well cared, then we're going to have this aggregation. And I'll tell you in the next few slides that aggregation is a trigger for immunogenicity. So the original biosimilar and the original drug and the biosimilar should be very well compared in terms of stability in order to have comparable immunogenicities. Just to give you 2 or 3 slides, I'm sorry to be so scientific at this point, but basically when the patient is having these drugs in infusion, the amount, if there is any problem here in the pharmacy or in the transport or the logistics, when there is this accumulation of immunocomplexes, there is this complex sizes that we need to understand that it comes to when it enters in the patients. And we know that, for example, I'm sorry, this basically, when you have this, basically these are immunocomplexes, okay? So when you have different immunocomplexes, this is small immunocomplexes, these are higher immunocomplexes. This means when it comes to the patient immediately responds with that, with hypersensitive reactions, infusion reactions. So when you have aggregated forms of a protein, then the patient will react immediately. That's what you see sometimes when there is this infusion reaction to drugs. Why is that? Because, for example, one of the things here on an infusion bag, when there is a filter here, and many times this question of the filter is very important. If the drug is not well formulated, if the quality of the drug is not very good, then there are aggregated forms of the drug that enters in the patient, and then the patient will react with this immune response against these immunocomplexes, and then we're going to have complement activation, immune cell activation, clearance by the spleen and the liver. So this immunocomplexes is basically what we don't want, and we need to compare that with the original drug and the biosimilar. This is very important because the immune system is so sensitive to detect these differences that if we implement the right techniques and the right clinical assessment, then we can compare the 2 drugs. So I'll go fast on this on the next slides, okay? So predicting the likelihood of immunogenicity. The companies know this. Why I'm telling you this? Because the companies have 20 years of experience. All these biosimilar companies that are coming to the market, they have experience on producing biologic drugs. And they know exactly what are the causes of that. For example, molecular structure, aggregates, change of primary sequence, fusion protein, everything that EMA and FDA asks for the quality of the drug really is to study what will happen in the patient in terms of immunogenicity at this point. The same, host cell proteins, host cell DNA, impurities. So the impurities, and it's very interesting now that FDA and EMA are so strong on this point that the sensitivity of detection of this aggregate, I'm sorry, I'm completely lost on this. When these companies are really studying these aggregated forms, these cryptic epitopes appearance, these modified amino acids, or when they go to this purity analysis, the sensitivity that the biosimilar companies are asking now to detect these differences is 1,000 times more than it was 10 years ago. So it's much more sensitive now to detect these little differences than it was 10 years ago. And it came with the biosimilars. So why aggregates are important for immunogenicity? Because they will respond vigorously. They think that aggregated forms are viruses. Okay? Think about a bad quality drug as a virus, and the virus enters in the human body, and then there will be a response. Okay? I will go over. So the mechanisms of immune response are T-dependent, T-independent, activation of innate immune modulators. I will not go on these mechanisms, but there is one important trigger. There is the signal 1, the first signal that is important for immunogenicity, which is different T-cell epitopes. And the biosimilars have exactly the same sequence as the original drugs. So the signal 1 for inducing activity and the trigger 1 is not present there. Okay? So, but then there is another trigger that is important here to trigger this activation of T cells, B cells, and memory B cells, activation of plasmacytes. And here we have some kind of inhibition of this activity. So if you have similar epitopes, there is another trigger which is important that can overcome this activation here, which is aggregated forms of a protein. Okay, so this is important. That's why there is this managing of product quality, regulatory approaches. And all these regulatory approaches are very important because they try at least to lower the risk of immunogenicity. So what is the risk of immunogenicity? Is the probability of developing a reaction times the severity, okay? And we have several conditions that are for lower probability of developing immunogenicity and higher probability of that. And here, the higher probability of developing immunogenicity is an autoimmune disease, okay? Comparing hematology with rheumatology, here the risk of developing, the probability of developing immune response is higher in autoimmune diseases. The chronic dosing, if it is a foreigner protein, of course, an antibody, infliximab or adalimumab is a foreigner protein, a subcutaneous impure aggregates. Okay, so this is very important for that to understand, and the severity, the same thing. Okay, so anyway, so what now that I gave you the basic rules of immunogenicity, so now we can go to the biosimilars and look the guidelines that European Medicines Agency and also the FDA have specifically for immunogenicity. So there is a specific guideline to assess how the immune system recognizes these different drugs. Okay, so all of these characteristics and points that I gave you before, it's very important for the regulators to dissect the immunogenic response against a drug. So there is this comparative assessment of these immune responses. I will not read this. This is on the guideline. You can keep this, the risk-based approach that provides a starting point to evaluate this immunogenicity with diversity risk factors. What is important is that the clinical trials don't be too much concentrated on a few characteristics to assess immunogenicity. Okay? And then the assessment is based on this evaluation of the evaluation of the final product in the treatment population. Okay? So now we can go. I'm sure that yesterday we have seen this triangle. Okay? So this is a triangle of the biosimilar development. This is becoming a classic in all presentations. But when you look at this triangle, basically all of this analytical testing are basically to eliminate any potential causes for immunogenicity in the patient. Okay? A major part of that is for that. So aggregated forms, exposition of cryptic epitopes, whatever. This analytical testing is very important for that. Not only for function, for the activity of the drug, but also for the immune activation that can get the drug can activate. So this is highly important. People think that this is more important than clinical trials. I don't see this triangle like that. I think the amount of information that you have here is probably more than you have here on clinical trials. But both of them, all of them are important. So when you have a drug, when you have a biosimilar being produced, and the clinical efficacy and safety we want to evaluate, in the middle of this, there is a lot of things. Okay? So the important point here for the last 20 years that we learned a lot, what is the cause of this immunogenicity? Okay? And if you look and if you see how these dots are connected, we know that all of them are connected to this production, but basically what is in the center of everything is the stability. Okay? So this is very important for developing immunogenicity because the stability should be very high. Okay? So I think that the companies, we have to ask the companies, and especially the hospital pharmacists, should ask a lot of data about stability of the original drug with the biosimilars. And I think we have good data for that, for clinical trials. But I'm sure that we need more data of these drugs in the market. Okay? These are two different things, because we know that companies make these variations in the process of in the process of production. So stability we need to understand how these biosimilar drugs will maintain stability in the market compared with the drug that was given during clinical trials. Because the process of production of a drug in clinical trials is not the same as when the drug is in the market. So that's why at the end of this presentation I will mention pharmacovigilance, and I think that pharmacovigilance is important because of this point here. So this is another way to put things anyway. So immunogenicity is on the top of our concerns. So anyway, I've told you this before, so amino acid sequence is the same, so there is no major interest on that. So there is a good assessment of all modifications of antibodies. This analysis is very strong. And one important thing here is the sensitivity. Compared to what we had in 1990, okay, 25 years ago, our sensitivity to detect these aggregated forms increased 10 million times. In the first antibody, when it came to the market, when infliximab came to the market, we did not have by far the same capacity to assess aggregated forms of a drug, okay? But we have it now. This is what we call mass spectrometry, okay? Mass spectrometry gives a good hint on that. So when a drug comes to the market, I think they have done a good job to assess that. And we need to have this assessment in the clinics, okay? So there is this stepwise assessment, and we come here to the clinical part now, when there is this uncertainty being reduced when we arrive here on the clinical studies. I'm not surprised that we don't see too many differences on the switch studies, because basically the analytical comparability was well done, but we need to maintain that in the market. I think this is important, and I think that this maintenance in terms of pharmacovigilance is very important. So on the clinical development, there are two things that are important. One is pharmacokinetics, and the other one is another one is immunogenicity. Why immunogenicity then? There is one theory, okay, on the switch studies, is that there is a carryover or epitope spreading that might occur when a patient moves to another, from one drug, from one batch of a drug to another batch. Okay? We don't have data for that. There's only a theory, okay? But it's a theory that now can be started to be assessed with the biosimilars. So this carryover effect from one drug to another due to immunogenicity leading to antibody formation. So this is a theory. Maybe it's not a true theory, but anyway, the regulators have this guidance to assess this epitope spreading. This is something that FDA is really cared of. Okay? They want to have good assessment on that. And then the practice. Okay, we have one bad case about immunogenicity, a strong case of immunogenicity with erythropoietin in 95-96. Okay, basically in 95-96 there was a changing formulation of erythropoietin, and then there is immunogenicity appearing here at this point when the patients were reacting against erythropoietin. Why? It was not a biosimilar erythropoietin, it was an original erythropoietin, because they changed the formulation. And when they changed the formulation of erythropoietin, patients reacted against aggregated forms of erythropoietin. Okay? So why this is important also in inflammatory diseases? Basically, here, because we have these citrullineated proteins that rheumatologists know, and citrullineated proteins are nothing more than less than aggregated forms of proteins. When you talk about citrullineated proteins or antibodies against citrullineated proteins, means that these citrullineated proteins are aggregated forms. I'm sorry, this is a normal protein, and these citrullineated forms of a protein, meaning that the patient sees these aggregated, rheumatological patients see these aggregated forms of a protein and responds against them. So patients in autoimmune inflammatory disease, in rheumatology, in gastroenterology, they respond to these aggregated forms. But for example, here when you compare rituximab, for example, and the immunogenicity of rituximab between lymphoma, patients with lymphoma and patients with a rheumatological condition, you see more response against rituximab in this condition than with lymphoma. Okay, meaning that there is a good the trigger of immunogenicity is also the disease. So what should we request for biosimilar clinical trials in terms of immunogenicity? I think the study should be powered to detect immunogenicity. Power meaning in terms of the amount of population and also the type of assay that's being used. The duration of the assay should be between 6 months and 1 year. Everything that is less than 1 year, I will not trust too much on that in terms of transient and sustained antibodies. Samples of the antibody samples should be taken when the circulating drug has cleared. So because there is an interference on the circulating drug on the immunogenicity assay, so the sample should be taken when the circulated drug is at a low level. Okay? The assay should be robust, sensitive, specific, and validated, of course, and the binding and neutralization antibodies should be measured. I mean, if you have all of that, if you have all of these conditions, I'm sure that we can trust on the data of immunogenicity. And going from the immunogenicity to the switch studies that was mentioned in the previous talk, basically what we see now is that when you look at the number of publications having switch data, we see a strong increase here more recently on switch studies. Okay? And it's interesting that all of these switch studies, at least half of them deal with immunogenicity. At least 50% of the studies on switch do not assess immunogenicity. It should be done. So there is a lack of information on those studies. If you look at, if you go at all of this data about immunogenicity on infliximab biosimilars compared to the original infliximab, adalimumab biosimilars compared to the original drugs, rituximab or etanercept. And when you look at, I'm sorry, this is not well formatted, but basically when you have these patients that maintain biosimilars and when they maintain here the original drug here on this light color, you see that there is a good comparability in all of these studies in terms of immunogenicity against infliximab, adalimumab, rituximab, and etanercept. Actually, it's very interesting for etanercept because we have some data. The data is not very conclusive, but we have some data showing that probably there is less immunogenicity against etanercept biosimilars compared to the original drugs. But anyway, the data is very conclusive that with all of these conditions about immunogenicity, we see very similar immune reactions. And we have done that. We have done more on that. And it's very interesting because when you look at samples from patients reacting against drugs, and we have done for infliximab, for example, we have here several patients, and we have here some different isotypes. It's not only IgG1 that respond against the drug. Several other isotypes also respond against a drug. So if the companies want to do a very good job on assessing immunogenicity, they should be concentrated not only on IgG1, but also in other different isotypes, for example, IgG4, because we know there is some variations in the type of antibodies that respond against a drug. The same for adalimumab. We see differences here on the type of antibody that respond against adalimumab in different conditions. So IgG4 is probably another step in the evaluation of immunogenicity. So another point here, the question of switch. When you have in the future, probably we don't want probably this, okay? But when we have this transition here, we need to assess very well if the immune system recognizes the drug, the original drug, and the biosimilar in the same way. So we know we have good data showing that the amount of antibodies reacting against the original drug and the biosimilar drug is basically the same. But the quality of those antibodies is the same or not, okay? So to evaluate this epitope spreading. So the important thing here when you look at an antibody, we need to understand and categorize, and this I think this is the final road to assess this immunogenicity, is to see if these antibodies that of a patient that react against different regions of the original drug and the biosimilar, if they recognize the same regions on one drug and the other drug. Okay? So this is a question that we try to answer. And basically what we got, also in collaboration with João Eurico, here was to, in opposition to what was mentioned before about the study of Ben Orin, a gastroenterologist, that he took serum from patients reacting against infliximab Remicade, and they took the serum from those patients and we went to see if there was a cross-reactivity against the biosimilar. So we did the opposite. Why we did the opposite? Because we wanted to understand if the patient have antibodies against different epitopes in the biosimilar compared to the original drug. So we did the opposite. So and the data really shows, and in terms of correlation, and the Pearson's correlation, we show a very good correlation between the serum that recognizes the biosimilar. It also recognizes different batches of the Remicade. So there is a good correlation. This is with 42 IBD patients. Okay, this was done in IBD patients. And basically we have shown that both in patients that were naive patients and patients that switched, there was a good correlation in the recognition of the biosimilar and the original drug. And the same for IgG4. So we went to categorize how much there was of immune response, not only with IgG1, but also with other isotypes. We went deeper on the dissecting the immunogenicity. And when we assess the different batches of Remicade and different batches of CTP13, we see that the antibodies IgG4 against infliximab were basically very similar. We went also to assess the capacity of those antibodies to neutralize CTP13 and Remicade. And when we look at these different batches of Remicade and different batches of biosimilar, we see that the capacity of the patients, IBD patients, that respond against the biosimilar, they have similar capacity to neutralize Remicade and similar capacity to neutralize the CTP13. So this recognition, this type of recognition was very similar between the two antibodies. And we went a little bit above that, which was to identify in the infliximab molecule where the antibodies from the IBD patients, which epitopes they recognize on the infliximab molecule, to see if the antibodies recognize similar epitopes or different epitopes. Okay? So we identified all these epitopes in the heavy chain of the antibody and in the light chain of the antibody. Why we have so many epitopes? Because this variable region is from mouse, and because they are from mouse, they have different sequences. That's why we have all these epitopes. But still we have some recognition of some epitopes here on the constant region. But to make a long story short, what I would like to that you see is that when you have this serum against anti-Remicade and anti-CTP13, basically when you have here the controls, you have here different epitopes, you see that patients react against the same epitopes between the two drugs. So patients with, that react against CTP13 or against Remicade, they don't see different epitopes on the two drugs. So this is an added proof that the immune system sees these two drugs exactly as the same. The same for the variable region, so there is a good assessment here on the type of epitopes. And we went also to isolate monoclonal antibodies, because this was with a polyclonal response in the previous section, but here is a monoclonal response. And we isolated monoclonal antibodies from these IBD patients, and we went to analyze if those monoclonal antibodies respond differently to different batches. And basically, we don't see differences, because when you have these antibodies here, these monoclonal antibodies here, they recognize monoclonal 1, monoclonal 2 against the constant regions. They recognize similarly the constant region, and here the same for the variable region. So at the level of the monoclonal, polyclonal response, for the immune system don't care too much. It's saying these two drugs almost as the same. So I'm almost finished, okay? So there is here the importance of immunogenicity for original biologics and biosimilar pharmacovigilance. Of course, now we have a guideline in Europe, a specific guideline for good pharmacovigilance practice. And pharmacovigilance practice really is also putting one emphasis here on immunogenicity. So immunogenicity is requested for the companies to continue to evaluate that in the market. Why that is important? Because stability of the drug is a trigger of immunogenicity, and we want to force the companies to assess these drugs in the market as the original drugs have not done it before. So this is very important. This is very new. This is the new guidelines of pharmacovigilance, and immunogenicity is a strong point on that. And of course, if you want to do a switch, of course, we need to assess all these questions here. Sorry. Of course, you need, when you do a switch study, of course, or a switch program, you need to decide on the follow-up, establish endpoints, but I think that immunogenicity and drug trough levels could be an added point here to assess the outcomes that come and to confirm the outcomes or not confirm the outcomes that come from a population of patients that switch to an original, to the biosimilar. So the conclusions are basically that immunogenicity testing rely on the design and execution of the assay. Okay, this is very important. I think we need to be very careful on that, and when we compare studies on the immunogenicity, when we say that immunogenicity is different or is equal or similar, we need to understand what kind of a test or assay was made and the characterization of that immune response. It is impossible to compare these rates of immunogenicity unless we have these head-to-head clinical trials, okay? And they should be really assessed at this point of comparison of the assays. And these head-to-head studies, I mean, these rates of immunogenicity should be well studied and well related to the state of the art, okay? And I think that I'll finish with this slide, basically to say that similar immunogenicity between the original and the biosimilar depend on the totality of evidence. So I've spoken on a few things, but I think what is important here at the end of the day is that we force at least the companies to maintain the same quality that they have proven in clinical trials. So thank you very much for